Eu vou tentar dar alguma formalidade para essa reunião, que na verdade é uma reunião de amigos, né, parceiros de trabalho de amigos. Eu vou fazer uma apresentação, primeiro em português e depois em inglês. Uma apresentação rápida. Então, bom dia a todos inicialmente. Eu sou a professora Carmen Lima. Eu vou usar as minhas anotações aqui, porque o número das instituições e de professores é relativamente grande. Eu sou da Oncologia Clínica da Faculdade de Ciências Médicas da Universidade Estadual de Campinas, da Unicamp. Eu tenho a satisfação imensa de receber o professor Ulrich Abram, do Instituto de Química e Bioquímica da Universidade de Berlim, para uma apresentação na nossa instituição. Essa apresentação ela vai constar de duas partes. Na primeira parte, o professor vai ministrar a palestra Abordagens teranósticas com isótopos radioativos. Esse é o futuro da medicina nuclear. E a seguir, uma segunda parte com perguntas e respostas sobre o tema apresentado. E essa sessão, então, vai ser presidida pelo professor Vitor Deflon, do Instituto de Química da Universidade de São Carlos, da Universidade de São Paulo, e com a presença aqui do professor Corbe, que vai nos auxiliar também nos questionamentos. Eu agradeço muito a presença de todos e agradeço em especial ao professor Ulrich pela sua visita na nossa instituição. Good morning. I am Professor Carmen Lima. I'm from the Clinical Oncology Service of the Faculty of Medical Science of State University of Campinas, Unicamp. I'm pleased to welcome the Professor Ulrich Abram from the Institute of Chemistry and Biochemistry at Berlin University for a presentation at our institution. The presentation will consist of two parts. The first one, Professor gives the lecture approaches with radioactive isotopos. Is it the future of nuclear medicine? Next, a second part, questions and answers on the topic presented, which will be led for Professor Vitor Deflon from the São Carlos Chemistry Institute of São Paulo University. And I would like to thank everyone for attending to this presentation and thank particularly Professor Ulrich Abram for his visit to our university. Please, Professor.
Thank you, Carmen, for this kind introduction. I'm afraid I cannot continue bilingual. Maybe German, English, but this would not help with this audience, I'm afraid. That's why let me try it with English. And it's bad style, I know it, to start with an apology, but I'm a chemist. And the content will be focused through the eyes of a chemist. So be a little patient when I show you too many structures, formulae, and so on. I try to shorten it, but sometimes chemists are strange persons. Look at me. Well, Theragnostic approaches with radioactive isotopes is in principle an old story, and I will try to summarize some of these issues, and I will spike it with some of our own results. We are not experts and we are not focused to Theragnostic approaches from the clinical point of view, but we did some research that finally led to such developments, sometimes expectedly, sometimes surprisingly. So let me first introduce the most important people that contributed to this talk, since these are the students and the colleagues who did the work that I have the honor to present here in Campinas. So let's start with the Frenchman Guillaume Claude. He is my last PhD student, and he finished his thesis this August, and is now working at the University Hospital in the Nuclear Medicine Department of Berlin at Charité. So we have a close connection also to the clinical side. This is a Campinas lady. Sue Ellen came from Campinas to make a PhD in our group a couple of years ago, and she was very successful. She made an impressive chemistry, which was at the beginning not intended to have any relationship to nuclear medicine, to application, but finally we find a bypass to this way. And I'm very happy to present these results of Sue Ellen here in Campinas. I think she made a master with Pedro Corby. So everything comes together. Also with this person, Pedro Ivo, who is in the audience. He also contributed to the translation to the clinical research, not to the clinical application of Sue Ellen's results when he was for a postdoc period in Berlin. He worked together with Anna, a master student of our university, and this is Sarah Spreckelmeier, the chemist in the university hospital in the nuclear medical department, who is responsible for the work there, and she also contributed to some of the results. Okay, it was important to me to start with these persons. To show them at the end is always that you have to hurry through the last pictures. They deserve to be introduced personally. Well, what is Theragnostics? At the moment, I have sometimes the impression it is a buzzword. It is used for almost everything, and no one has really a content, and so I ask the institution that knows everything, Wikipedia. So this is the definition of Theragnostics you find in the English version of Wikipedia. And Theragnostics is an emerging field in precision medicine that combines diagnostic and therapeutic approaches to provide the potential of personalized treatment and real-time monitoring of the effectiveness of treatments. Well, in principle, this is something that physicians do since centuries. Diagnostics and therapy, and using both together. This is nothing extremely new. And of course, this definition has not been by a bot. It has been taken from a paper that has been published in the European Journal of Nuclear Medicine and Medical Imaging. So there are also names behind these definitions. And I, in principle, completely agree. But since Hippocrates, you do that, diagnostics and therapeutics. So you want to help the people and need information from the diagnostics. So far, so good. What might be new is the real-time monitoring. And this is really a help to make it personalized, not overdose the patient. I think this is a real thing that this is remarkable. Let's go back. Let's go back to a time where everything began from the chemical point of view. And this was in the 30s of the last century. I think it was 1936 when the therapeutic application of radioactive iodine isotopes came into the discussion. Not iodine-131 that we use today for treatment of different diseases of the thyroid gland. It was the idea born by Saul Hertz, who had heard a talk of a physicist, interesting, a metaphysician, physician heard a talk of a physicist, and he came to the question, couldn't it be to produce a radioactive iodine isotope that helps us to treat cancer, to help patients? And he asked this question in 36, and the physicists could help. They provided him with a radioactive iodine isotope, not that we use today, a completely useless isotope from our present point of view, iodine-138, but it could be made that time. And this is also the context in which I want to regard our own results. Not everything that we do today will come in the same way to a practical transformation to the patient, but it's important to start with something. So he started with this by far too short-living isotope. It has been produced in a machine which was 10 kilometers away from the clinic site. That means only on the way to the clinic site, one half-life was away. But he could make the first animal experiments with iodine-138, and 2 years later, also 125 and 130 were available, not 131. Why? Since it could not be produced in an easy way in a cyclotron. That means it took 10 more years until the war has finished and the nuclear reactor that have been developed for a nuclear weapon came available for useful things for the production of radioactive isotopes. And in 46 and 46, the production of 131 iodine started in the United States. And today, 131 iodine is a standard agent that is used for treatment of different thyroid diseases. The result of this development, and this is what we can learn from this process, was a strong collaboration between physicians and physicists. Chemists were not so required that time since there was no chemical challenge to make iodide, which is finally the source for this treatment. Nevertheless, a chemist was involved. This was Clancy Seaborg, but that time he worked as a nuclear physicist and made nuclear reaction. Both of the chemists have of the physicists have big names. Enrico Fermi was involved and Clancy Seaborg was involved. Both received the Nobel Prize a couple of years later for other work, not for the iodine work, but they had an eye also to the transfer into the clinic site. Remarkable is the Seaborg picture. I have one of his latest years. This is the first person that ever has been honored with the name of a chemical element. For you, it may be the same, but for a chemist, it would be a big honor that one of the elements has your name. And Clancy Seaborg was still alive when Seaborgium entered the position in the periodic table. A curiosity, now we have another example from a Russian or Georgian nuclear chemist, Oganessian, who has also been honored in the same way. The problem is, radiation is not always so dangerous as we think. He became older and older. And what they feared is that no more elements would be left before he died. That's why they decided, I think it was a good decision then, since Seaborg contributed to many of nuclear research. How iodine works in thyroxine is in principle clear. So we all have heard about this. We know that these thyroxines contain 3 or 4 iodine atoms, and they work independent of the isotope that is included. Our body, or the body of most of the mammalians, have a big drive to accumulate all the iodine, which is in principle rare element, into this function, since the thyroid gland is responsible for many, many functions in the body. And so very effective mechanisms have been involved to couple these thyroid hormones to their binding sites. Also, this is something that we can apply. That's why I show it for our thinking about theragnostic accumulation of some chemicals to specific sites in the body. We have, in principle, this lock and key, a way where the shape of a molecule fits to a shape that is preformed at the biological site. An example for thyroxine is this trans-thyroxine, which is a protein that is formed by a tetrameric unit, so we have 4 almost equal chains of protein, of peptides. And this molecule, when we turn it a little around, has a hole, or has 2 holes preformed to welcome these thyroxine hormones. So when we zoom a little inside this picture, then we will see that there's already a thyroxine inside. So we see it, but it's not yet highlighted. And when we show this receptor-bound thyroxine, then we see that the iodine atoms exactly fit the hole that has been preformed by nature to welcome them. So this is a principle that we should apply when we make some new pharmaceuticals from which we expect to bind to some receptors in the cell or better on the surface of a cell. This was iodine, and this diagnostic and therapeutic connection just with one and the same element is in principle an exception, or was that time an exception, from the simple conclusion that there are not so many radioactive isotopes available from the same element that time that could be produced by a relatively cheap procedure. Since this is the other side, when we want to apply something, there must be someone who pays for the story. That's why when we speak about Theragnostics, we normally think in terms of two elements, two similar elements in the best meaning of this word. So probably here in the house it is used, a combination of gallium and lutetium, probably also indium and yttrium. They are so-called matched pairs, which have the same charge at the atom, at the ion, and sometimes we believe that this is enough to make a matched pair. This might be not completely right. What do I mean? Let's start with the good news. It works, and it helps many patients worldwide, this concept. And everything that works is good. Since I'm not just a chemist, I'm a potential patient. So I also have to provide this view to the story. So a combination of a matched pair is fine when it helps to treat the cancer that has been detected by the diagnostic isotope. So the first example that I show is the DOTA-TOC combination of indium and yttrium, but it also works fine with gallium and lutetium. And what is applied from the chemical point of view, a big molecule, is this combination of DOTA and TOC. TOC is a not so big peptide, and DOTA is just a name for this chelator where we want to bind a radioactive isotope, a metal ion, in the center. So there's one border between the active site that is responsible for the biological targeting of the conjugate, and there's the other side that is responsible for the binding, the tight binding. It should not be released in the blood flow, for example, during the application. This is DOTA. And in some papers, and to be honest, I suffer when I see that, we have this depiction. The radioactive isotope is somewhere in the middle of this tetradentate ring of the nitrogen atoms, and then we have the conjugate, and it works. Well, it works, and it works in such proper way that no one really cares what is about the radioactive isotope, since it is simply the same. So modifications are normally done with the peptide sites, since this allows different accumulation under different circumstances in the patient. This is important, and this has been done chemically by the modification of these peptide sites. So we come from DOTA-TOC to DOTA-TATE. When we make a small modification in this side chain and to a relative new DOTA-NOC, we come by a modification of this phenolic group by a bicyclic aromatic building block. So nothing extra. Optimization of the biological properties are done by the modification of the targeting unit. This is fine, and this is what makes it working. This is, with other words, sorry for the coordination chemist, the most important site of the DOTA-TOC conjugate. But what is with the rest, and why do I suffer? It is not so easy to say the metal is somewhere here in the middle, since the metal, and any metal needs different environments, has to be coordinated. So when we use gallium at this place, we need not just these 4 atoms, but two more. And we have it available here in the side chains. These carboxylic acids can be deprotonate. They can bind with two of these positions to the gallium. And then we have a positively charged complex. Here's no TOC bonded, as you see, at the moment. With lutetium, this is bigger, we have another situation. It established a complex with a coordination number of 9. That means lutetium does not just take these four nitrogen atoms, not just 2 more carboxylic acids, it needs all 4 carboxylic acids. So when we look to the pure lutetium complex, it is an anion and completely different to the gallium one. Nevertheless, we work both. And there's another big problem with the lutetium. When we want to bind TOC, we need one of these side chains. This is no problem with gallium, since 2 of them are left, so we can use one of these 2 to bind to TOC. But here, with lutetium, we have in principle nothing available. It should not work. Should, but it does. Why does it work? Since one of these bonds most probably is cleaved by combination with TOC, and then we have another, maybe neutral complex of lutetium, where the empty coordination position of the carboxylic acid is replaced by water. So we are in a very lucky situation, and despite the fact that the chemistry would say this cannot work, it works. And this is good. Why it doesn't work, you can simply see from the ionic radii. We have this tiny, small gallium with a radius of 62 picometers and almost a double radius for lutetium. So it is hard to expect that they bind in the same bonding site of a chelator. And as we have seen by crystal structure studies, they do not. Here, the driving force, why it works with gallium, indium, yttrium, and lutetium is encoded in this biological part, in this biochemical part, in this peptide site of the TOC conjugate. This conjugate this part has so a high affinity to the somatostatin receptors of a cancer cell that it does not care about what is in the other bonding site of the molecule. This is not necessarily the case in other examples. So we must be aware that it only works because of this high affinity. But as long as it works, it's fine. So we can say biochemistry is important, the peptide. Coordination chemistry, at least in these examples, has a minor role. It must work, it must bind tightly, but how it bonds does not play a significant role. And the same is with the PSMA receptor binding site. This conjugate works in the same way very effectively with gallium, with lutetium. It is in application in 2 different versions. I think this is the new one, which is not yet approved completely, but it works also very well. And this part binds very effectively to the membrane-specific antigen of the prostata, and so this makes the molecule working, irregarding the chemistry that it's enclosed. And nowadays, there exists for PSMA a lot of examples. It works with gallium, it works with lutetium. There are some agents also available for technetium. Where is something available, and this is a nice idea, to remove all this binding site and to label the PSMA itself with fluorine 18, and then we have a relabeling situation where we have just to exchange one of the atoms and have in principle copy of the bioactive part of the molecule. So the matched pair concept works well with these 2 pairs, and it should work also with rhenium and technetium. Rhenium exists in form of a generator, nuclide 188 for therapy, and technetium-99m is the long-working workhorse of diagnostic nuclear medicines. There are worldwide 40 million administrations per year. So technetium makes the main job in the practical side of nuclear medicine. Nevertheless, it would be good to have the congener with this for the therapy. Chemically, they are a kind of siblings. They belong to the same group in the periodic table, from which chemists sometimes conclude that chemistry should be the same. This is not the case, not the same. The chemistry is similar, but the differences are big enough that this causes problems by a simple transformation of a technetium solution to a rhenium solution. This has two reasons. One is that they have different redox potential. That means oxidation, reduction is not the same for technetium and rhenium. And the kinetics, the speed in which a reaction works, is not the same. And also this influences the products. Another problem, what we sometimes forget, is technetium pharmaceuticals for diagnostic are, in the most cases, single molecule solutions. That means they have been developed for one organ. They have been made for brain perfusion, for myocardium perfusion, for kidney function studies. And so this cannot be generalized. And the other point is the targeting has specific effects, and there's in principle also no exact need to double it with rhenium, even when it works. And this we can see with the technetium MDP, which is used for bone diagnostics, but there's an equivalent for rhenium established for a long time, and it works also with a phosphonate, but with another one. It would work with MDP as well, the rhenium solution, but there's no need since the MDP works good enough. And so again, what works in the clinical side practically without problems, why shall we change it? So let's find or let's look for new solutions. So here this doesn't work, but it works with some other examples very well. The redox potentials come apparent when we go to the newer, the modern kits. The old kits, maybe the MDP kit, has a lot of reducing agent. This tin fluoride is good for the reduction of pertechnetate to this pharmaceutical here. And this is a lot that is contained. So simply, it's the same if we take technetium or rhenium. It's enough also to reduce rhenium. With HMPAO, with this brain kit, it was necessary to make a very well-balanced amount of the reducing agent, since only a rhenium technetium 5 complex was desired. And any over-reduction reduced the accumulation in the brain. So here, only a small amount, this amount that is necessary, is contained. And this small amount is not good for work with rhenium. But we don't need it. We don't need a therapeutic element and therapeutic drug for brain perfusion. We would need it for a glioblastoma, for example. But for this neutral membrane-penetrating agent is not good to work. So that means it is not always easy to transfer technetium to a neighborhood who has a nuclear reactor in his neighborhood is really encouraged to do this, but for a mass production with a close interaction with the clinical site, with a transfer to the clinical application, I think we are decades away from real serious application for a significant amount of patients. But it's an interesting point also for the therapeutic chemistry. Let's come back to the to the definition. Theragnostic is an emergent field, And here I want to highlight the imaging technique, the improvements, and the combination with targeted therapies. When we think this bigger, and you have done it here in Campinas, as I could see 1 hour before, then we should go to other combinations. Okay, the standard things I spoke about was nuclear medical imaging with nuclear medical therapy. This is what we have with this mixed pair concept. Why not, and you did it, include CT, MRT, even elastography, any other good working, well-established imaging technique with a nuclear medical therapy. This would deliver progress immediately and not in 10 years time scale. Why not using nuclear medical imaging with chemotherapy together? This is not new, this idea. It is in principle used since the 30s of the last century with the iodine, but freshly considered. Let's think solutions which did not work properly 50 years ago. The techniques have improved, the isotope production has improved, so think new if something is on this path that can be used for a new approach. Why not using nuclear medical imaging with nuclear medical therapy and chemotherapy? Sounds strange, I will show you an example. And why not considering modern targeting strategies that makes the use of different isotopes much more comprehensive and much more easy to combine when we have drug delivery systems where we can mix inside 2 different isotopes. This is a good approach, but the reality is a little far away, but it's worth to take some research to this. Let me show you one example with the chemotherapy. It's an old molecule. This is fluorouracil, and it has been introduced since 60 years as an established drug for different kinds of cancer. Why not using it together with fluorine-18 as a Theragnostic unit? Then you have the real molecule, the same molecule, without changing any component for diagnostics. Okay, this idea seemed to me good, but it's by far not new. This has been developed decades before. And the idea is so straightforward that, of course, the people had them. So in '82, the first studies with radiolabeled fluorouracil have been done. And even in '89 here, used as prognostic agent, so the terms of Theragnostic was not yet developed, but prognostic, this was exactly the idea. We should use the one-to-one molecule for therapy and diagnostic. And it did not come to reality from a simple reason. Fluorine-18 chemistry is not so easy as it appears to be. Fluorine chemistry is an extreme chemistry, and particularly when you have some aromatic rings, they cannot so easily be substituted with fluorine-18. You don't have the compounds, you don't have a big amount of radioactivity available for this. Everything works well like FDG, where you can start with the fluorine minus anion. But when you use some electrophilic reagents, it doesn't work so easily. But a lot of progress has been done. Why not thinking it newly? And another point is fluorine atoms have entered the classical pharmacy. So this is simply a statistic about fluorine-containing drugs which have been approved by the FDA during the last decades. And we see from the 2 decades, from 2000 to 2020, more drugs have been developed than in the time before, containing fluorine atoms. These are not just therapeutics, oncologic compounds, but let's have a look to these compounds. If some of this can be prepared easily by fluorine-18. But for this you need a cyclotron and you need a fluorine staff and a fluorine lab. But it's worth that since a big market is underway for this. Well, a number of different reviews that describe the role of non-radioactive fluorinated compounds and describe also the progress in fluorine-18 radiochemistry. A lot of solutions have been found for non-radioactive syntheses. Not all of them work up to now for fluorine-18, but no one invested the time to develop it. Also, this would be worth to consider. The remaining problem is the half-life of the isotope. This we cannot change. The distribution logistic, you should have it close to the clinics. And we end finally at individual molecular solutions. So any synthesis is an individual synthesis. You can make it in the batch operation, then you have 5 or 10. This would be good for a clinic site. And I saw the synthesis apparatuses in your research lab. This can be programmed in a way that even not so complicated molecules can be synthesized, but the price per batch will be maybe high, but also with some of the other solutions. But then let's go to an alchemistic approach. Sorry that I bother you with too much chemistry, but I must introduce the chemistry of Sue Ellen, who comes from Campinas. And this is really alchemy. What Sue Ellen did during her PhD work in Berlin, she used the gold complex, a simple one that has only function to deliver the gold atoms. The chloride will be removed, the sulfur-containing molecule will be removed, and gold remains to react with this not so easy, but also not too complicated molecule. And then she mixed that in one pot and added other metals from the periodic table. Any colored metal has been used and has been studied in such reaction. These are seriously one-pot reactions. She mixed it together. I could order chemicals from her. I said, well, and I'm interested in the iron complex. Then she said, okay, I go to the lab. Next morning, she came with crystals, with single crystals of a pure compound. So this chemistry is really a simple chemistry by self-organization. So the forces from this ligand and the gold direct the molecules. So when I show you this mixture, I should divide it a little. She has 3 families of products obtained, depending where these elements are found in the periodic table. The one is very simple with alkaline earth metals, strontium, calcium, barium. She has such a ring system consisting of 4 gold ligand units. They form a ring. In the middle, concentrated, are 2 of the green metals. When she moved to the other metals of the transition metal side, copper, cobalt, manganese, nickel, mercury, she had 2 of the gold units combined with 1 of the metal. And then she used 3 plus metal ions with 3 positive charges, with 1 exception. She obtained the same structural feature, irregarding which metal it was, with 1 exception. 3 gold atoms in a ring system with 3 of the ligands, and the center, 1 of these pink elements. All these compounds are characterized, all the other prepared, and when we look back to the periodic table, we meet some old friends from nuclear medicine. We meet here lutetium, we need gallium, indium, yttrium. We need samarium, holmium, erbium, terbium. So all these work with this strange molecular approach. Okay, not pharmaceuticals, chemical compounds, but combination of different metals. And to be honest, when Sue Ellen was in Berlin, we never had a thought in application for nuclear medicine, since it seemed, at least for me, too far away from any reality. And then came the next step with these persons here. I introduced that in the beginning. And last year, we had an exotic idea. since besides some other isotopes in reviews, you always find gold-198 as a potential isotope for nuclear medical therapy. Maybe this is driven by some persons who like the gold chemistry, Sylvia Jurison. Maybe in Missouri has been done a lot of this work. Gold-198 is a beta emitter with, in principle, suitable half-life and also with a good energy for therapy. And it can be made by neutron radiation of gold-197, the normal gold. The normal gold has only, the gold in our rings has only one isotope. The back side of the story, or the bad side of the story, is we will never have the chance to produce gold-198 non-carrier added. We will also have the standard gold isotope in the solution. So we make a kind of labeling. And this is not good for a therapeutic approach, since we carry a lot of gold of a metal through the pharmaceutical, which would be useless. And it should not be useless, and this is what I mean, to combine with therapeutically interesting metal ions. And gold is a good therapeutical ion, and from the chemotherapeutical way of view, and then we can combine radioactive gold with toxic, cytotoxic gold, with indium, even with lutetium, then we would have a therapy-only molecule, or with gallium or mixtures of indium and gallium. This would require a biological activity of gold as from the chemical side. Then we would have a double third of gold, radioactivity and cytotoxicity, cancer-static action of the molecule of the gold compound. And this is not excluded when you look to the literature. Of course, this auranofin is for a long time used as a bad agent against arthritis. But also in the last 10 years, there was found some anti-tumor activities, and nowadays you find a couple of reviews that describe auranofin as good therapeutic drug against some different cancer sorts. So that's why here are some recent reviews about gold, the application of gold compounds, and many more. So there's not space to collect them all here, but Google can help you to find some suitable more reviews. But I must highlight, of course, that of Petro Ivo in the first one here. So this is the end of the story. Hopefully not. This is the beginning of the nuclear medical story. So I'm a little happy that it's finally accepted, the first paper of this group of chemists, Anna Baitulina, Guilhem, Suellen, and Petro Ivo, and Sarah as authors of this first nuclear medical paper that has been published recently, last week, that combines gold-198 with lutetium and gallium mixtures, and also describes some of the biological activity of the non-radioactive gold compounds which are required to justify this work, to continue this work. Well, and when I should also have a few words to modern targeting techniques, and when you want to go deeper into these details, just read the review of your colleagues from Sao Paulo. I think they are from Araraquara. This group has written a review where approaches, where nanoparticle-based solutions, where liposome-based solutions are reviewed. And the idea behind is when we can immobilize radioactive drugs of different isotopes in one of these nanoparticles, then we just have one problem, the standard problem of nanochemistry to target. So this is not yet a solution, but it is also an approach that we should think when we think into therapeutic pathways. When we go away from the complicated chemistry to a simple chemistry, and we succeed to target the cancer by the surface of a nanoparticle, then we can include the radioactivity of both jobs, diagnostic and therapy, into the particle and transport it to the position of action. This might be easier with the classical nanoparticle approaches than with liposomes, since liposomes are biologically active molecules, and we did some liposome work 20 years ago, and we were simply not successful. We could concentrate in the middle of these liposomes radioactive compounds, but on the way to the body, in the body, they decompose simply, and then they spread out their activity, and we found it everywhere. But also here might be true, we can go to next generation of drugs, and so let's inspect what is the progress of the liposome in my cell chemistry in the last 20 years. So what's the conclusion? I have no conclusion that is comprehensive, so I take one from an extra issue of bioconjugate chemistry. More chemistry is needed for molecular imaging, and we can extend it, and therapy. What does it mean for chemists? The nuclear chemistry should be considered in order to make new isotopes accessible. So cyclotron chemistry, reactor chemistry must be checked what is possible and what can be reasonably produced there of interesting isotopes. And the demands must come from the clinical side. You should not just just claim, I want to have a good isotope. You should say, is it possible to produce that, or that isotope? What will it cost? Which amount can you produce? So make these demands, since these demands may encourage people for research. The technical nuclear chemistry might be optimized. New generator systems should be available. We learned it with the gallium-68 generator. The first generation was lousy, but now they have improved, and now they live the time that they should physically work. The first generation broke down after half of the possible times. So there is space for improvement. Organic chemistry must be developed to make efficient labeling routes. Coordination chemistry must be improved, and organometallic chemistry must be improved to develop nucleators. Biochemists are required for the development of efficient coupling techniques to the biomolecules. They must find out what is the molecule, what is the part of the bioconjugate that we should produce to target a cancer cell with a high yield. They were successful with PSMA. Why not with other cancers? And we need an efficient drug delivery, not in the small percentage that we are used to have from the diagnostic point of view. This can be solved, in my opinion, only by a tight collaboration of different branches. Let me say, as you do, physicians, chemists, no, chemists should not be second, physicists, biochemists, biologists, and chemists. They should work together, they should learn to communicate together. This is what was my experience with the clinical sites. We use the same words, but we mean completely different things. So we need a kind of translation of the terms used in the clinics and used in the chemistry lab to come to a really fruitful communication. It will work, I know that it will work. Yes, and finally, we need collaboration in the analysis and solution of the problems, since only together solutions can be found that one branch defines as a problem. Do it together, you do it together. So my talk was useless here, since you all did the things that I wanted to suggest. Nevertheless, I enjoyed to stay here, and I hope I didn't bother you too much with chemistry. And the persons, the important persons, I showed you in the beginning, the not so important institutions, but finally without them we could not do the work since they give the money. Are mentioned here. They are Brazilian institutions together with German institutions, and in this isocyanide chemistry also the University of San Diego, the University of California in San Diego was involved. So I can end, almost end, with the story, and probably I try to convince you that chemistry is not just boring work. I was inspired by your Christmas tree, and I copied this in the last minute to this presentation. This is a Christmas lecture that we make every year for the students and also for some school classes who come to the university. Then they all want to study chemistry, and they see the chemistry day by day is not the same like this show. May I ask not to put this into the YouTube presentation since the music is not free. So thank you very much for coming. I hope I could help you with the last presentation a little over the first hour in which I have bothered you. Thank you very, very much.
I'd like to thank you very much for your brilliant talk. And now I'm going to invite Professor Victor Deflon for the second section.
So, good morning. I'd like to thank you, Professor Abram, for so interesting presentation, so nice talk, very impressive results, and very nice video also at the end. We have some time for discussion. He showed us how interdisciplinary is this work. It's very important for us to discuss and to try to understand each other. So I would like to answer the CancerThera project that enabled this moment, and also the program from the faculty that enables this program, and Professor Ernesto that came together with Professor Abram, and that they spend this time to come to be here with us. So I would like to give you then the opportunity to make some questions to discuss with Professor Abram.
Thank you, Professor Abram, for a wonderful presentation. The end is the show. At the end it was very nice, but it is a very wide lecture with fits exactly what we intend to do in our project in CancerThera. I would have many questions. I'm not a chemistry, but I have a specific question about the pair technetium rhenium and the molecule sestamib or tetraphosphine, which we use for myocardial perfusion. But we know that sestamib does not uptake only in the myocardium, but also in tumors, different tumors. We published sestamib uptake in multiple myeloma, and sometimes with uptake much higher than in the myocardium. So is it possible to label sestamib with rhenium to use the pair for teranósticas?
I'm pretty sure it will work. I've never tried it myself, but we did the chemistry of our contribution with the substituted ones together with rhenium and with technetium. And they both end in the hexakis complexes. It simply, no, you don't have a generator. It would simply be the opportunity. I can ask in Berlin that the colleagues take a kit of sestamib and use 188 rhenium for one experiment, then we will see from the HPLC what is the product. I'm pretty sure it will work, or it will not be a big deal to prepare a kit that works with sestamibie or with other glioblastomas. With the MyoView, we came to this point from a project that we have together with our colleagues, where we should find some diagnostics, and we say also therapeutics for glioblastomas. And also there, this cationic myocardium agent always shows some activity. And so we tested it, and this works with MyoView. We simply took some kits and added 188 rhenium instead of 99M technetium. And there's also a paper from Indonesian groups. I've cited here in the presentation. I can send it on my computer. This works quite fine. With sestamibie, I don't know. I'm pretty sure it will work. There were some papers about this. I do not know what is the accumulation rate, since sestamibie is finally not made for this. It has been made for the myocardium. But from this base, when you find the accumulation, it would be worth to substitute, since they have prepared for the development of sestamibie, I think 20 and more isocyanides. We should go back to the roots where they accumulate. And sestamibie was best for myocardium. Maybe others are best for something else. The synthesis of these ligands is not easy. They are stinky liquids. And that's why in the kit is the copper complex. So they did not think in terms of click reactions or anything else. They simply needed a way to provide a ligand in a form that can be lyophilized to make a kit. But I'm pretty sure you will find something.
Thank you. I have another question about alpha emitters. We see people are using actinium-225 for prostate cancer and other cancers, but it's very expensive and it's not available, easily available. But radium-223 is much more available. You can buy radiochloride, which is used for treating bone metastases. What do you think about using radium chemistry to label peptides, for example, to treat for theranostic use?
This is a promising pathway, is a promising idea, and no one did it in the past in reality, since the chemistry of these second group elements was not so interesting for anything. So you can play with the barium chemistry, and what is good for barium should also bind to radium. And the chelator chemistry is simply not developed, or at least underdeveloped. And there should be something possible simply to look to the chemistry of barium, which organic molecules binds tightly to barium. Another point that some conservations that I have with alpha emitters is that they will destroy most probably organic skeletons, since the alpha radiation is that rich in energy that they will destroy carbon-carbon bonds. So even when you make a nice labeling, you must be fast with the distribution before the self-destruction mechanism of the molecule happens. I do not know about the energies of this radium isotope, but they found this with astatine that was in the 80s or 90s under discussion for some application in therapy. And finally, they were successful whenever they could avoid organic molecules. And radium is used, as far as I know, as a nitrate or the chloride at the moment, and not in form of an organic chelator. But simply use it, try it. We can check if ligand is suitable, and in vitro you will find how stable is this potential drug. It depends on the alpha energies. When you're lucky, it works. If not, not. Since in principle, the actinium story, they use PSMA, and they use also DOTA. So they use organic ligands. So it might work, maybe not good enough, but alphas are difficult. I see it from the patient's point of view.
Thank you very much.
Oh, I can stand. When I fall down, you put me up.
Okay. Yes, more questions? Simone?
Good morning, I guess. I'm a medical physics. I got my undergraduate last year, and I'm doing research in Alzheimer's disease. And I would like to know if about the opportunities of a PhD in neuroscience, nuclear medicine applied to neuroscience and neurodegenerative disease. If you know a little bit like some opportunities for students, I saw that at the beginning of the presentation. You had some students that came from Brazil and from Campinas, and I want to know if, how is it like to a student like me can go there and make like research and other things.
You must find a suitable group. So I'm not suitable in terms that I'm retired. But to go to Germany to make a research, a PhD or whatever, is not a big deal. So we are very international. So I was almost not German in the row of the students I talked about today. But this is standard in our group, or was standard in our group. We have people from all nations and from all continents. And so as a Brazilian in Berlin, you will not be an extraordinary person. You will be one under millions, no, thousands. So this is not a problem, but to find a suitable group is another point where you can make exactly that work that you intended to do. I can send you some email addresses of colleagues who work related to this nuclear medical physics, since we work in a collaboration project with some of these colleagues, and they are closer to the physics. So I'm a chemist. So you probably would not be happy with me.
No, you're a really nice guy. Thank you.
Congratulations for the excellent presentation. My question is about the spacer. What do you think is important to have a spacer between chelator, agent, and molecule?
This is what we learned step by step. We first made a conjugation without additional spaces, and then we learned also the nature of the spaces is not the same. So we now end with some lysine spaces, 3 or 4 lysines that we can simply include in the peptide synthesis. This works well, but also some ether groups coming from these polyethers work well. So not each spacer is a good spacer, and unfortunately we did not find that solution for each of the of the peptide. It's different.
It's difficult. And another question is about the future. What do you think about the future in teranóstica?
A simple answer: the future will be brilliant. And the second part of the sentence is when we work hard enough. It's hard to predict. So all these potential solutions have their justification. And it's the question what you want. You can hunt some easy solutions, then you should start with easy molecules. Let me say, I showed you one molecule in the beginning, this MAC3. This MAC3 kit contains finally, for its activity as is needed, a free carboxylic group. Chemically, this is an invitation to use it as coupling a peptide to this position. Then it's no more good for kidneys, but might be for other organs. And this is an easy solution. This is a clear solution. And in principle, you can find out the results within the next 6 months. What I presented here with these complicated isocyanides and also with the Suellen results with container molecules, this is in the very beginning. This counts in years. But there are some chemical solutions that already work. MAC3 is one of them. And why not modify it? Since chemically everything is prepared. And this compound is even easy to synthesize. Simply consider to make it. And then a simple test will show you if the conjugate is stable enough. You need from the peptide size a spacer, and then you couple to the protein as an amide coupling. This is standard. It should work.
Thank you.
But probably it's oversimplified. But without trying, we will never find if it works.
More questions?
Good morning. Without considering the high price, I want to know what limitation do you consider to be the most important when you start to work with isotopes in limitations? What the most limitation?
The isotopes are the limitation. The price comes from the availability or from the bad availability of the isotopes. You must be able to produce all these isotopes, unluckily, at the same time when you want to use it at the same time. So last year I was to a conference and these people from the Paul-Scherrer Institute in Switzerland, they gave 5 or 6 talks about terbium. This is amazing. It works wonderful, but it can only be prepared there at this point, at this site, since they have the equipment. So on the other hand, when you find a solution that works excellent, then there will be some companies who make it possible to produce it for a more reasonable price. So both sides might work. So the economic side is not strictly related to an impossibility of the use. Finally, also, the iodine story I mentioned in the beginning started with a completely unsuitable isotope, and it ended with the solution of iodine 131, which is applied until today. And even the technetium story, I didn't tell it today. It would be an overload of technetium. It has been developed in Brookhaven, and they wanted to take a patent for a technetium generator, and some economic departments have refused to patent a technetium generator since they make the prognosis this is good for nothing. And they lost billions of dollars with this decision. This was good for the community since the generator was free. Anyone could make it. But it was simply a misinterpretation of the situation. This was in the 50s. It was not even economically wrong what they did. But always to look only to the economics might be not a good solution. When you find a chemical, a physical, a medicinal application that is worth to spend money, you will find the money. Lutetium is also not cheap.
Thank you.
More questions?
Okay, it's not a question, but a complete the answer for the student. I think the important is to have access to radioisotopes. This is a problem, really. And technician is easy. Professor Dario has a generator in the hospital. He has a lot of has. Then you told about to have more chemistry. I think you need to have more radiochemistry too. Then the idea is to get students in biomedicine, chemistry, pharmacy. You put to work in technician chemistry. It's a fantastic chemistry. Several oxidation states, several chelators. Then before to thinking copper, something gallium or others, get students to training in technician chemistry, to do a chromatography, to labeling. Then it is an idea. It's in my mind, I have students that we work in fluorine and the gallium chemistry, but I put them in the bench to work in technician first, labeling, chromatography, system counter, to learn about how radiochemistry works in nuclear medicine or in chemistry department too. I don't know what you think about this.
I'm the wrong person to have another opinion, since I make technetium chemistry myself for 40 years. You cannot expect from me that I say technetium chemistry is boring and not worth to be done. But you're right. On the other hand, technetium chemistry is from itself a little complicated. What you mean with this tracer chemistry, this should work. This is what we wanted to implement for a decade in our lab course, to use a standard kit, to let the student prepare the kit, to make the quality analysis by HPLC, by chromatography, to see what can happen, which mistakes can I make during the synthesis or during the analysis, simply to get access to radiochemical procedures, which are not theoretical procedures, which are applied in reality. We finally failed in the money for buying the HPLCs for the lab course. That's why we did some paper chromatography and not more.
Professor, first of all, I would like to thank you very much for coming for this brilliant conference. And as you know, we are starting our project, and we have 10 years, actually 11 years to develop this project, supported by our research agency of São Paulo State. It's a big responsibility. Me are retiring as you as well. In 2 years I have to leave officially, but I intend to develop this project, and I would like to thank you. I would like to thank all the people that work in this project. It's a multidisciplinary, it's a multi-professional. We believe in it, and at the end, I would like to invite you to join us in this project if you want, if you have time to, and for us it's very important if you help us, because we have to choose the best ways and the right ways, and we know that research has some risk, and for us it is completely new, and oncology, I believe it's probably will be a good way in the oncology in the future. Mm-hmm. We have a lot of patients without therapies, without possibilities, and this could be a possibility to treat patients in the future. And so I would like, on behalf of our group, thank you very much for staying with us and to coming in Campinas. I know that São Carlos, the students are better than Campinas, but no, I'm teasing you. Thank you very much.
I never said this.
You have seen so nice results of swelling that he showed.
Swell is from Campinas. No.
She was previously a student of, no, it's not in this group, no. But she was originally from the chemistry institute here and went to Professor Abram to do the PhD. I would like to thank Professor Abram again. I would like to say I have some questions, but I can discuss with him later, including the chemistry. I just would give some words about the presentation. He showed us how much work is to be done, and that is very nice for us. There is a lot of things to do. You can improve everything, every stage that he showed, every step, the bioconjugation, the chelation, the targeting, and biodistribution, everything. You can see very nice systems that he showed us. And we can also see that sometimes we plan something, we think if we have more sites for bioconjugation, it would be good. And then we see that it's not the case. We need to do the work to have these answers. And when we see a system that works well with lutetium and with gallium, then we see the structures. We see that we can do better. Even if, despite the fact this working, it's well, but it can be better. We can do everything better if we work in these projects. So if you would like to do some things to close about that, I would like to thank you very much for coming. And you also for coming, for having this moment and these discussions.
I have to thank for this invitation. It's always nice to be in Campinas. It's my third time. I have remembered. First I was just with Pedro Corby, then we made some promotion for Ciências Sem Fronteiras a couple of years ago in chemistry, and now it's the first time in the medicine department. So thank you very much for this invitation. I enjoyed the visit here. I still enjoy it, and I hope for better weather for the weekend, since we want to go to the beach. Thanks.
Okay, thank you.
Neighborhood who has a nuclear reactor in his neighborhood is really encouraged to do this, but for a mass production with a close interaction